Hi everyone, this is Kevin Wagner, the Keto Advocate. In January of 2016, I had the privilege to sit down and chat with Dr. Adam Hartman at the first annual conference on nutritional ketosis and metabolic therapeutics held in Tampa, Florida. Let's see what Dr. Hartman had to say.
My name is Adam Hartman. I'm a pediatric neurologist at Johns Hopkins Hospital in Baltimore. I'm an associate professor of pediatrics and neurology at the School of Medicine, and I have a joint appointment in the Department of Molecular Microbiology and Immunology at the School of Public Health. In my practice, I see patients who have primarily medically intractable epilepsy, which means they haven't responded to the first couple of medicines that we use. And therefore, their options include surgery or metabolism-based therapy. I also have a laboratory where we're investigating different aspects of the ketogenic diet and other ketogenic substrates in seizure management.
Could you tell us about the topic you'll be speaking about here at the conference?
So the topic I was asked to discuss is about the anti-epileptogenic mechanisms of the ketogenic diet. And it turns out this is a pretty complicated question. The way that we're going to break this down is into the acute anti-seizure effect versus a more chronic anti-epileptogenic effect. And by that we mean that whatever the treatment is that we've selected to study is going to address the underlying pathological processes that lead to the development of spontaneous recurrent seizures, both in animal models and in people. It's an important distinction because we haven't really made any headway with the medicines that we've used in the last 50 years. So the medicines we use now are no more efficacious than they were 50 years ago. The medicines now have fewer side effects and fewer interactions with other medicines, which is important. And they're better tolerated, generally speaking, than the other medicines are. However, we still have a pretty significant gap of patients whose seizures are not adequately treated. In adult populations, it's about a third of the population. In pediatric populations, it's about 20%. So we're still talking about a substantial number, and this is a population that's utilizing quite a bit of medications. They have a lot of involvement from the families in terms of making the children's environment safe, making sure that the impact of their seizures is minimized in terms of physical effects. And the medications themselves have a number of side effects as well. So what we ultimately as a community are hoping to do, we've sort of done a decent job treating seizures acutely. What we now hope to do is turn our focus more on the chronic picture. And so in my talk, we're going to focus primarily on the long-term picture, looking at some human data, looking at some animal experimental data, and then we're going to start looking at some of the published mechanisms themselves. Finally, we're going to talk about how our quest to finding the anti-epileptogenic mechanism has actually led us to identify a number of novel treatments and re-identify some older treatments for treating seizures as well. So my lab is currently looking at the impact of the ketogenic diet as a comparison to intermittent fasting. And so a number of years ago, we published data looking at the impact of intermittent fasting in mouse models. So the ketogenic diet was designed to mimic the biochemical effects of fasting. And it would stand to reason that the mechanisms of fasting and the ketogenic diet supplementation would be identical. And we actually showed that in a number of mouse models, they were not only not identical, they were actually opposite to one another. And so that led us to study intermittent fasting further. One of my colleagues, Eric Kossoff, and I have published a small case series looking at the efficacy of an intermittent fasting regimen in children. And what we found was that there was some limited efficacy in a number of the patients, although it clearly was not a home run, but it's worthy of further study and investigation to try to optimize the fasting schedule that these children had. We also are doing studies looking at the impact of the mTOR pathway, which is a master metabolism regulator, and in trying to dissect the biochemical precursors to activation and inhibition of that pathway, we've actually found that the ketogenic amino acid L-Leucine, the L isomer of leucine, which is commonly used by bodybuilders and people who want to supplement their amino acids actually has anti-seizure effects in animal models. That's not necessarily new news. Some data, similar data were published back in the 90s. But the impact in one of the seizure tests, the canic acid test, which is an excitotoxicity test, was positive, and that was unexpected. And so we then looked for a possibility of a contaminant or transporter-mediated effect of the L-leucine, and in our studies we used D-Leucine, which is the D-isomer, and as it turns out, was found back in 1906 by none other than Dr. Emden, who the Emden-Meyerhof pathway is named after, glycolysis. Dr. Emden published in German in 1906 a paper looking at the ability of L versus D-leucine to produce ketone bodies, and it turns out D-leucine produces more ketone bodies per a given unit than L-leucine does. Interestingly, we didn't see an elevated level of beta-hydroxybutyrate in the blood of the animals that were being supplemented either with D or with L-leucine. But we're continuing our studies of D-leucine, again shifting our focus from the more acute models, which we published last summer, to the more chronic models of epilepsy. And thus far we have some interesting preliminary findings, but we're not quite ready to fully discuss that yet. So I think one of the most interesting studies that came out recently was from a group in France looking at changes in metabolism prior to the onset, just prior to the onset of seizure-like activity in cultured hippocampi, intact hippocampi. So the hippocampus is a part of the brain that is generates a lot of seizure activity, and this group was able to culture them intact, and so all of the basic wiring within the hippocampus was maintained, although the external connections obviously were cut. And what they showed is that there's actually an increase in oxidative phosphorylation just prior to the onset of the electrical activity changes. In one of the models, they used bicuculin, which is an inhibitory neurotransmitter antagonist, and so it enhances excitatory activity. And through their studies, they found that indeed there are also chronic after that decrease in oxidative phosphorylation with the persistence of the seizure-like activity, which in a human, if you looked at electrical tracing like that, you would call it a seizure. They show that there was actually a decrease over time in oxidative phosphorylation. And interestingly, oxygen utilization in those cultures, they were able to monitor the two at the same time, turned out to have a mirror response, which really reinforced some of those findings. So I think one area that is very interesting from an acute anti-seizure perspective is whether we can impact that pre-epileptiform change from a metabolism perspective. A lot of us have suspected for a long time that metabolism is important for seizures, but most of the evidence for that came from studies looking at impaired mitochondrial function from surgical specimens or from animal models. And although that was important work, and obviously multiple studies looking at the utility of the ketogenic diet in treating epilepsy are also important, there was one critical piece to that line of logic that was missing, and this study now fills in that one missing link. So the next question that's going to be interesting to see is what happens when you alter the culture media to have more of a ketone body type of an environment as opposed to glucose. These were young hippocampi obtained from young animals, and so they were still in a ketone adapted state. So it would be interesting, and they were maintained primarily in a glucose-containing medium, so it would be interesting to see what happens if you were to alter the medium. There's a whole variety of other experiments that can be done with that. But it also has implications for the more chronic story that I mentioned earlier, where there may be metabolic perturbations that are chronic and fluctuating, and then there's some threshold that's crossed. And after that threshold is crossed, you develop seizure activity. So I think that very short time frame just prior to the onset of the epileptiform activity in the culture is a critical thing for us to study. It's critically important not only because the group that did this noted the changes. The next logical question is, can you alter something about that change? Can you prevent that change and therefore prevent seizures? Can you use that change in metabolism somehow to identify a target and exploit that molecular target to then prevent seizures from recurring? Not only occurring, but then recurring on their own. So I think there's a lot of very exciting work that still needs to be done. The question of what happens to ATP during seizures is an interesting one. And I'm not sure that all of the data line up to a single answer. So in the study that I just mentioned with the cultured intact hippocampi, the group also showed that with more chronic self-generating activity, which would be similar to what we see with status epilepticus, so really prolonged seizure activity, that the energy stores actually appeared to be fairly intact and improved over time. And so the question of metabolic failure or energy failure during status epilepticus, which is something that a lot of us have taught and thought was actually happening, are actually not supported by at least this most recent study. And so I think we don't have a phenomenal idea of what exactly is happening with ATP production. I think that this study brings some of the older data into question, and I'm not quite sure where. So I think it's important to be studied so that we can get some more solid answers on that. Supplementing ATP. So ATP has a number of different potential actions too, although it's an energy source, obviously. It also is a precursor for other compounds. So I think it may have transporter effects as well. So I think there's an awful lot to learn about ATP during seizures and whether levels change over time. One of the problems with ATP is that its measurement can be very inconsistent and very sensitive to the conditions under which they were made. And so there's a lot more to learn about ATP, and I think the methods that we have now are probably insufficient to really get a grasp on that, but hopefully over time that's going to improve. So this is one of the classic questions about the ketogenic diet. So the way I think of it is in three different levels. The first is individual substrates that contribute to the effect. So is it low glucose? Is it increased concentrations of ketone bodies? Another way to look at that, which is a little more complicated, but is a little, I think, closer to the actual situation. If you alter one component of a diet, you have to alter another component to make up for that calorically. And so that was part of our thought process behind the intermittent fasting study. The third level of complexity would be looking at pathways. And so this sort of follows our work over the years. And that's how we ended up studying the mTOR pathway. And when you look at regulation of the pathways, it can be either one of those that may perturb that pathway. And so I'm not quite sure. When you look at low glucose conditions in culture, Susan Messino has some nice data. Looking at the impact of that, I don't know what the impact was if you were to supplement those those media with ketone bodies, what would change about her data. And so I think I'm not quite sure how to experimentally separate the two. There's a body of work from the folks at University of Wisconsin looking at 2-deoxyglucose, which is a glycolysis inhibitor and a hexokinase inhibitor, and that appears to have anti-seizure and probably anti-epileptogenic effects as well. When you talk to the folks who did that work, I think their general sense is that that probably is not the only thing that's involved in the ketogenic diet. There's probably more to it, which would support looking at diets as a whole or at pathways in terms of trying to figure out what's going on with the ketogenic diet, which I think I'll go on record saying I think it has multiple mechanisms, but that's not really different than a lot of the drugs that we use. Very few of the drugs that we use, particularly a number of the newer ones, have a single mechanism or a single binding site. Some of them do, but many of them have an impact on multiple neurotransmitter systems, and so I don't think that the diet is necessarily, any of the diets are necessarily different than the drugs with respect to having a single versus multiple mechanisms. So the mTOR pathway is a master cellular metabolism regulator, nutrient sensor, nutrient integrator. And mutations in that pathway are seen in some human diseases. And so the most straightforward way to explain this is that the proteins that are responsible for the upstream regulation of that pathway put a brake on cellular growth. And if you're missing important parts of that brake, it's sort of like not having the pedal and you just have the brake shoes. And so if you're missing that, you won't be able to activate the brake. And those syndromes tend to be so-called overgrowth syndromes. And so in tuberous sclerosis, which is a mutation in either TSC1 or TSC2, you see overgrowth of, you have tumors, you also have other tissue abnormalities called tubers, and a variety of other abnormalities. There's also syndrome associated with other, a number of other proteins that are upstream even of TSC1 and TSC2. I don't think that we have a fantastic idea yet of how that then converts into seizure activity, except to say that abnormalities in that pathway lead to abnormalities in connections, in dendrites, and you can reverse some of those abnormalities by inhibiting the pathway. So Mike Wong at WashU and his group showed, Sharon McDaniel was the first author on that paper, showed that the ketogenic diet inhibits the mTOR pathway. And we actually had an unpublished finding just before that paper came out showing the same thing. Interestingly, the intermittent fasting model did not inhibit mTOR pathway activity, which also supports the idea that fasting and ketogenic diet have different effects. But then we did some follow-on work looking at the acute anti-seizure effects of rapamycin, which is an mTOR pathway inhibitor, comparing it in a number of seizure tests to the ketogenic diet. And what we found, surprisingly, is that the rapamycin is only weak in inhibiting acutely induced seizures, and it works in a test where the ketogenic diet is ineffective, and the test where the electrical test where the ketogenic diet is most effective, and we tried a variety of different dosing schedules for the rapamycin, but we couldn't get it with any of our dosing schedules to work in that particular test. So our view is that although the mTOR pathway is inhibited with the ketogenic diet, we don't think that that explains the acute anti-seizure effects. We don't really know what the impact is on long-term seizures with respect to the mTOR pathway, and so that's work that hopefully someone's going to do. I don't think we have the full answer to that question. So there's the paper from Japan looking at the inhibition of vesicular glutamate release with acetoacetate, and I'm not sure that that the answer for the other ketone bodies has been fully derived yet, particularly with respect to acetone. So we have some unpublished data looking at the supplementation of a ketogenic diet with L-leucine in the drinking water. And what we actually found is that the L-leucine decreases the efficacy of the ketogenic diet. And so that's just in a single animal model. We also looked at another ketogenic amino acid in our publication and found that it was not effective in our main test, the canic acid status epilepticus test. We also have some unpublished data looking at the other branched chain amino acids, and their impact was statistically but not clinically meaningful. So the scores over time were different, but they didn't have that sudden drop-off sort of midway through the monitoring period that we saw with L-leucine. So I think that that's something that needs to be investigated further in other models of seizures in animals. You know, the branched chain amino acids also have some toxicity. There's a disease called maple syrup urine disease, and high levels of leucine and probably one of the other two also That leads to a substantial amount of central nervous system toxicity. So we need to be very careful. Fortunately, we found that the doses needed for the effects in our test were very low, but I think we need to be mindful that too much leucine is probably a bad thing. Leucine has a number of different effects. It has a number of transporter effects. It has a number of endocrine effects. So I think that, which have not been seen with D-Leucine, and so I think that our efforts are really focusing on what we, at least at this point, perceive as being the safer isomer. So we had a paper published last summer in Neurobiology of Disease looking at the impact of D-Leucine on two different types of seizure tests. One was the canic acid model. So this is an excitotoxicity model that leads acutely to status epilepticus, and we found that D-Leucine terminated the status epilepticus at a wider dosing range than L-leucine did. We also showed that it inhibited seizures, or elevated the amount of current needed to generate seizures in an electroshock model, the 6 Hertz test. So we were unable to identify a definitive binding target, but it also, at modestly high doses, also appears to have an impact on neuronal excitability in cell culture. So we're pursuing studies in more chronic models of epilepsy because that's really where the clinical need is right now.